My name is Laura Weinstein, and I'm a rising sophomore here at DiD, majoring in biomedical engineering. My research this summer was on improving the purity of DiD-Encapsulated nanoparticles by Triton-Washing. Nanoparticles have extensive applications in modern medicine, the most recent being the Pfizer and Moderna COVID-19 vaccines. In order to research efficacy, it's first important to know where the nanoparticles are. To do this, fluorescent dyes such as DiD are often encapsulated within the particles as a way of tracking them. This summer, we engineered polymer-based nanoparticles of DiD for tracking. There's never a 100% encapsulation rate, so it's standard procedure to filter out excess dye from the solution using a filter vial that retains nanoparticles but does not retain DiD. However, we were able to notice that none of the excess DiD was actually filtering out, because if it was, the flow-through would be blue, but as shown in figure 2, it's clear. We also tried to filter the solution using a size exclusion chromatography column, but the nanoparticles and the dye move together through the gel beads as one homologous substance, when the excess dye should have moved slower than the nanoparticles. Lastly, when testing the cell uptake of nanoparticles, the plot of the DiD nanoparticles had a similar shape to the DiD alone control group, confirming that there was excess DiD in the nanoparticle solution. All of this led to the conclusion that the excess DiD is forming micelles, which are nanoparticle-like structures that only contain dye. Those micelles are unable to be filtered out using traditional filtration methods and can lead to false measurements and downstream experiments. To solve this problem, we found that adding the common surfactant Triton X100 for the filtration step led to the disassembly of the micelles and allowed for the dye to be filtered out. As shown in figures 6 and 7, the flow-through after the inclusion of Triton is blue, indicating that DiD is being filtered from the solution of nanoparticles. To further analyze this process, we measured the size and zeta potential for the nanoparticles, shown in figures 8 and 9, which are common steps in studying nanoparticle stability. The similarities in size and charge give us insight into why this is such an unacknowledged problem. The micelles effectively hide in the solution from these standard measurements. Additionally, these results indicate that Triton wash does not negatively impact the size or surface charge of the particles themselves. Returning to analyzing the cell uptake, the drop in fluorescence intensity in figure 10 suggests that the DiD has been removed from the solution. Meanwhile, the shape of the histogram in figure 11 for the Triton-washed nanoparticles is Gaussian, which is the characteristic shape of successful nanoparticle uptake. Together, this collection of data suggests that the DiD micelles have been removed from the solutions. Right now, all of our cell uptake data has been at a 1-hour time point, but in the future, we'd like to explore many time points in order to accurately show how DiD interactions are affected by the Triton-wash method. Overall, DiD encapsulation is a popular method of tracking nanoparticles. However, I have shown that this method creates micelles that need to be removed, and that our Triton-wash method is able to do that, including a detergent wash step will improve the accuracy of nanoparticle tracking and solidify the results of any experiments utilizing DiD nanoparticles. Thank you.